you can see, this is a first webinar about organ on a chip and Flamin-go project. And I'm super happy today to have the lead of the project, Dr. Annalisa Chiocchetti, with me, with whom I will talk all things about immunology, organ on a chip, and maybe a little bit about the future of medicine, because I guess this is a thing that's going to be important for all of us, for our health and our treatments in the future. This is a first part of 6 webinars that we will do in the next 2 months with different researchers that are part of the Flamin-go project. And first of all, let me just quickly introduce Annalisa. Annalisa is a professor of immunology from University of Piedmont in Italy, and she's the lead on the project Flamin-go. Otherwise, she has a really, really big expertise in immunology, especially in autoimmune diseases, and she's an MD and also has a PhD. She's very much focused on research. She also teaches and is an author of more than 75 scientific publications, as well as has 3 patents. And to start with, so I'm not going to be boring you with this long introduction. Annalisa, you are an immunologist. How did you find yourself in this field? Where does the interest to go into immunology come from?
Thank you very much, Seria, for this invitation. I'm very happy to be here and to share the Flamin-go project with you. Actually, why I'm in immunology is a long story. It has been a long journey in the sense that I started, let's say, at the age of 5, 6 years old, thinking and saying that I want to become a medical doctor. And when I grew up, every time someone was asking me, you know, what do you want to do when you will be big? And I was saying, okay, I want to be a medical doctor. And then when I reached the age and I could go to university, I wanted to be a medical doctor, but unfortunately, my parents were changing town, and so we had a big mess. And so the first year I couldn't go and inscribe to medicine. And so I decided, okay, just not to lose a year, I would do biology, you know, because some exams are the same. And so after the first year, I was there saying, but, you know, studying 6 years and then to specialize, and then it's a long, long time. And so I changed up my mind and I decided I want to be a biologist. And so I had a PhD in biology. But then, you know, actually, I wanted to be a medical doctor, you know, to deal with diseases, but it was impossible at that time in my mind. And so I decided to go to agriculture, because there I could study in a department of vegetal pathology. And so I started to study fungi that are pathogen for some cultivar, you know, and it was really, really fascinating for me. And I went to doing a postdoc on this topic in Paris, okay? And unfortunately, that year I had a very nice project because the aim was to identify which gene is responsible for the pathogenicity, okay, of these fungi. And so I really worked a lot for 1 year, but unfortunately, I had no results at all, no results. And so it was not possible because our experiments were done well, but I had no results. And so we sent back the fungi to Italy saying, but are they really pathogen? And they told me, oh, we made a mistake. We gave you a non-pathogen. And so I worked for 1 year for nothing at all. And I was 30, and I thought that my life was end there because, you know, after 1 year, I had no lab to go in and nowhere to go. And that's why I went to have, you know, a chat with a friend of mine in Novara, because in Novara, that is in the north of Italy, there is this new medical university. It was a small one. And so I said, okay, I will have a look there. And there I met, you know, a professor in immunology that would have become my mentor. And he proposed me a beautiful project that was to identify a gene associated with autoimmune diseases. And then I fell in love with immunology, and actually I became an immunologist.
Oh, that's a really interesting story. It's also quite encouraging for all young researchers or scientists that might listen, because I think everyone every now and then finds themselves in like, oh, what am I doing? Something is not working when you do science. So this is like, it's really nice to hear.
It's really, yeah, it's true.
That you had that kind of experience as well. So in the last 2 years, I work also as a science journalist, and I was covering pandemic quite a lot. So we all learned somehow. And there is this quote from a famous science journalist, Ed Young, who said, like, basically immune system is just really complicated. And I feel quite inspired that you are trying to put a little bit of it on a chip, which is like, seems like a really a crazy idea. There definitely still are a lot of things that we don't understand about immune system. And what parts, you already mentioned a little bit, that you focused on a specific gene connected to some diseases, do you focus on in your research work?
Yeah, let's say that you are right. He was right. Immunology is complicated. The immune system is complicated. The reason, if you give me 2 minutes, I just try to explain why it's so complicated, you know, because all our body is done of different organs, and each of these organs has got a function, okay, and it is connected to the other. So we know that, I don't know, the liver has some function, the heart has different function, and so on, okay. Each of these organs are composed by different cells, okay, very specialized cell. The immune system is exactly the same, is an organ, and is composed by cells that are specialized for their function. These cells are white blood cells in the blood. You know that the blood has got red blood cells, platelets, and then white blood cells. So when we say white blood cells, leukocytes, we refer to the cells of the immune system. But unfortunately, there are lots of, unfortunately, no, but I mean, the complicated thing is that there are several different cells within the immune system. So basically, they do different things, and also the same cell can do different things depending on which are the cells in the surrounding, how is the body going on. And what we know now of the immune system is really, really a little. So one fascinating thing of being an immunologist is that you've got lots of things to discover. You know, it's a really emerging field, as we are on the tip of the iceberg, let's say. So coming to my work, I work on autoimmune diseases, which are diseases of the immune system, meaning that somehow the immune system makes some mistakes. Usually the immune system is there to protect our body from pathogen, you know, viruses, bacteria, fungi, but sometimes it makes a mistake. And in autoimmune disease, the mistake is that instead of recognizing pathogen, it recognizes our tissue. And so it starts aggressing, like if it was an infection. And so this makes damage in our body. Okay.
And about one of these kind of diseases we will talk later. But first, I just want to quickly introduce Flamin-go project. This is financed from European Horizon 2020 projects. It's one of the projects focusing on health. And I think it has quite a big potential to be important organ on a chip, as well as just this part of Flamin-go for the future of medicine. Could you just tell us a little bit what is actually the Flamin-go project? What are you proposing? And
Yeah, sure. If I can, I will just share my screen with you. I prepared a couple of slides. Can you see my screen?
Perfect.
Okay.
Perfect.
So let's say before going to the Flamin-go project, I just wanted to introduce my center, the place where I work. So we already said that is in Novara, in Piedmont, which is in the north of Italy. Okay. And this is my center. It's a center for translational research on autoimmune and allergic disease. Now, I want to present the center for this reason. This center has been conceived looking at the big centers on cancer, because we have improved a lot in knowing immunology and also pathogenicity of the cancer, because a big center like this has been built. And what is the characteristic of this center? Is that they put together different realities. So in my center, we've got a block with the clinics. So obviously it is devoted to autoimmune and allergic disease, but patients, instead of going into the hospital, they come into our center to meet the doctors. Then we've got a second floor in which there are the wet lab where we do the research. So we have the samples of the patient. This is stocked into our next generation biobank. So all the samples are stored there, not only the sample, let's say blood sample or biopsies, but also all the information of the patient. That day that I had that sample, were you sick, were you okay, what was going on, which was the therapy, and so on. And so we've got the wet lab, and now I will just tell you two things on the wet lab. And then in the first floor, we've got the business incubator, because, and this is one of the things that European projects are really devoted to, is once you find some novelties doing research, you need to take them to the market. Otherwise, you know, we discover a lot of things, we publish a nice paper, but then that stop. And this is not a way to taking really to having an impact from your results. Now, let's say that our center is devoted to study the patient samples with a genomic approach. So let's say now it's a bit complicated. It's just to show different instrumentation and facilities we have that enable to study in a very on frontiers, let's say, way, the samples of the patient in a way that in a hospital, obviously, you cannot study. Now, coming to our Flamin-go, because the question actually was about what is the Flamin-go project? And
yeah, exactly.
Yeah. So I decided to put this is the Flamin-go of our, is the logo of our project. As you may see, Flamin-go, okay, it's a bird, of course. But I also put the title because the title somehow can be informative. Let's say that Flamin-go is the acronym from pathobiology to synovia on chip, driving rheumatoid arthritis to the precision medicine goal. Now, here we see a Flamin-go. He's got a problem in his joint because this joint is inflamed. And actually, our acronym wants to mean inflammation that goes away. So Flamin-go. Now, obviously, to describe the project, we need to first know what is rheumatoid arthritis and what is precision medicine somehow. So I just want to make a short introduction on what—
This is exactly my next question.
Okay.
What is rheumatoid arthritis?
Yeah, so let's say that it is an autoimmune disease. It is a situation in which we've got a chronic inflammation of the joint, so what we usually call arthritis. Now, if we have a look to this picture, we can see a joint. So a joint is where two different bones come together. These bones are connected by a synovial membrane, which is a thin membrane of cells that are there for one specific reason, meaning to secrete into the joint the synovial fluid, which is a lubricant that will allow the movement without pain. Okay, this is the normal condition. What happens into rheumatoid arthritis? It happens that the immune system, by making a mistake, starts to aggress the synovial membrane. And so it starts an inflammation that causes these cells to proliferate a lot. And you see in the right part of this drawing, you can see this big inflammation with a lot of cells that are producing lots of inflammatory mediators. Okay, and so the patient will feel pain, will have a swollen joint, but since it is chronic, as the diseases progress, this inflammation starts to erode the cartilage and the bone, and so will take, as you see in this part of the picture, to the deviation of the joint. And so people will suffer not only pain, not only swollen joint, but also a deformation. Now, this is rheumatoid arthritis. There are several, let's call them unmet needs. Flamin-go wants to address especially one of these. So let me try to explain how a patient goes through this disease, because first of all, it takes a long time to get the diagnosis, because it's not easy. We don't have a biomarker that say, okay, if you have this, you will have rheumatoid arthritis. And so this takes time, and during this time that I don't know it is a rheumatoid arthritis, this inflammation goes on. Then once you have the diagnosis, you need to have a treatment, of course. On the market, there are lots of different drugs that can let's say, treat those kind of conditions, but they are effective. But they are effective only in a small part of the population with rheumatoid arthritis. And unfortunately, at the moment, we don't know, we don't have biomarkers saying this patient will respond to this treatment. So how do we manage this disease? So patient has a diagnosis, and every patient will start by using methotrexate, okay? On these patients, average 40% of them will not respond, which means that after 6 months, they will go back to the rheumatologist and say, okay, I still have pain and I still have a swollen joint. So methotrexate is not okay, but 6 months have passed. So we go to a second line treatment. And on this second line, 40% of that patient will not respond. So this is to try to think that some patients succeed immediately, but some other starts cycling from one drug to another until they find. And some of them, there is a small proportion, let's say 15–20% of the total, who have no treatment at all. This means that we cannot stop the disease, because just one last thing, we cannot cure rheumatoid arthritis with these drugs, but if we select the good one immediately, we are able to stop the disease. So the disease do not progress, and the patients feel better.
Because in this way, if you're using so many medication, you may be mitigating the disease, but it doesn't stop it, and even mitigation, it's probably not really strong. Yeah, this is you can see how big impacting can have on people's life. And do you have maybe numbers of how many of patients with rheumatoid arthritis are there, like let's say in Europe or maybe in Italy?
Yeah, there are at least, let's say that is 1% average of population. It's 3 million in Europe. So it's a big problem.
So I think now it's clear why you focused on this disease with such a unsuccessful treatment currently. But what do you want to improve, and how can you improve this?
So let's say that there are lots of, the scientific community is trying to address all these unmet needs with what we usually call precision medicine. So let me just make a point of where we are now. Let's say that we wanted to develop a new drug for rheumatoid arthritis. We've got evidence that maybe some molecule can be a good target, okay? We develop it. And how can we prove if this is useful or not? Usually we start by using cells in culture. Cells in culture, for example, synovial cells, in this case, put in culture, and we treat them with the drug. This is very easy to do, but obviously cells in culture are not complex enough to recapitulate what happens really into the joint. And so to go to a more complex and physiologic situation, we go to animal models, what we call preclinical models, because in this way, the joints are really the joint, they are complicated, there is the immune system and so on, and we can treat. But obviously, animals are not humans, so it is more complex as a experimental model, but it's not usual. And so at the end, we go to what we call clinical trial. This is a randomized clinical trial. So I take 100 patients with rheumatoid arthritis. I divide it into two groups. Now here I've got the healthy subject just to test if it's toxic. In any case, I take 100 patients, I divide it into two groups, one I treat and one I don't treat. What is the results of this testing? What I said before. So let's say that among these, 60% will respond to the drug, but there is a part that will not respond. If you see in my drawing, there are different colors for each of these patients because each of them has got a sort of different rheumatoid arthritis, okay? So what is the aim of the scientific community in this moment? Is to go to precision medicine. What is precision medicine? Is that I don't treat patients like if they were all the same, but I study these patients very, very deep. So I take their samples and, for example, I make genomics, which means that I take their DNA and I study and I sequence all the genes that are inside to see what is different among them. This is one. And there I can do transcriptomics. So let's have a look to how the cell work and which kind of protein or RNA they express. And then by putting all this information to, and then I can also add information on the lifestyle, on the food and how they eat and all those stuff. So I take big data, big information, I put them all together, and at the end, I will come out with saying that this population is made by black, and then there is a second population made by green, and then by orange, and then by yellow. And for each of them, I will have a right treatment. So precision medicine is I study in deep the patients to give to each their own treatment. We all believe that this will be successful and that we will arrive probably in 10 or 20 years to really have drugs that are effective because we are able to identify those biomarkers that are important. So the Flamin-go project is something different because in my mind, in 20 years, we will be here. But we don't have 20 years. We've got lots of patients that are that need some solution now. And so the aim of the Flamin-go project is a little bit different. And so it says, okay, I will not go, I know that I've got black, I've got green, and I've got yellow patients, but I don't go into details searching for some difference. But instead, I try to make a culture of themselves. So I take the cells, biopsies from the patient. So each patient will have its own cells. And then I culture them in this chip. Since these chips are very small, I can make from a small biopsies a lot of different chip. And so I can test in the same time, in the same moment, all the drugs that I've got on the market. Just to give you an idea, if the Flamin-go will succeed, we can, if I take a biopsy today from a patient, within 1 month, I will have tested all the drugs in the market and I can say, okay, this is a green patient, I need to give the green drug.
And 1 month is enough for like from biopsy to growing cells on the organ on a chip and then already to seeing effect from drugs?
This is what we expect from, and this is what we are planning to do in this proposal. Yes, because organ on chip are solutions that are able to answer to all these. So they give complexity, but they are to do. Let's say.
And if I just go a little bit more into the details about organ on a chip, we are talking about organ. We are not just talking about cells, right? This is special. What is special about it? Like, why is it organ? Yeah.
Okay, so let's go to what organ on chip are. So I put some slides just to show you what we are talking about. Unfortunately, I don't have our chip now to show because I would have liked to show you. But in any case, these are different chips in the that you can find in the literature. So it's a very small device. And as you said, actually, it is a cell culture. So the objective is to culture cells. But when we usually culture cell, we culture them in a 2D monolayer, which means that all the cells are attached to a plate, one next to the other. But this is not physiological, because no matter what organ, the cells are distributed in a 3D configuration. Not only they are in a 3D configuration, but they also connected with other cells that compose an organ. So let's say that when you put cells in culture, you put one cell type. When you want to make an organ, you need to put different cell types, okay? And so the idea of organ on chip is exactly this, is to try to put different cells that are within an organ into the same culture, not in a monolayer, but as they are in vivo. So we don't want to make the same shape of the organ, but we want that the physiology is there. Now, since I know it's complicated, when I started to write the Flamin-go, I had no idea what organ on chip. I prepared the slide that up to me is really, really interesting, and it is explicative. This is one of the first organ on chip that has been done, and the idea is to make an organ on chip for lungs. Now, the organ on chip is done in this way, so it has nothing to do with the shape of the lung. But now I will show you how, by making this, no, it doesn't work. Why doesn't it work? Oh no, there is a video here. Unfortunately, it is not going. This is a pity. Anyway.
We will share it in the comment under the video.
Okay.
Afterwards, for anyone who is interested.
Okay. But just to explain you here, what they've done, you can see it here. There was a membrane, a porous membrane, and there were two different cell types. One were the lung cells, the other one were blood cells, okay? Because this is what happens in the lung. You know, you breathe, and so the air comes in, and then the oxygen needs to go to the bloodstream, okay? And so what they did is exactly the same. These two different chambers. In one there was the air, and in the other one there was the blood. And this was relevant for what you can. And moreover, there was also a vacuum, as you may see here, that can stretch this cell, because it's what, when you breathe, you make stress to the cells in the lung, and then when you breathe again, so there was a pressure inside, mimicking exactly what happens when you breathe. And this was relevant not only to study the physiology of breathing, but also because in the air you can put bacteria or you can put viruses, and in the blood, and so you can study how the blood cells can go to recognize the pathogen, aggress them, and so on. I really am sorry that you cannot see, because it was self-explicative, and it was really interesting to see.
I'm sure our listeners and viewers will watch it later. It's really, really an interesting.
So let's say that. Oh no, it's going.
It's showing now. Yeah.
Okay. Probably I just so this is how it's done. Now it shows you in a section. You see there are two. There is a membrane here in which you can see the cells, and you can see them on top, and it will be epithelial cell of the lung, and you can also put them on the bottom, and is the cell of the vessel, you know. And then there is this vacuum channel that mimics the breathing, and so this makes it's an organ, because there is both the alveoli and also the blood, with all the cells inside. There was air passing like when you breathe, and there is this stretch. And the application, let's say, of this organ are different, because as it is shown now, if you flux bacteria within the air, these bacteria will attach, as it does when you have an infection, and white blood cell will sense this bacterial and will start to extravasate, go and recognize, and this allows you not only to culture cell, but to culture them in a relevant way, like if it was the organ. Okay, so let's say that on the market there are lots of different organs on chip, so that have been done to understand either physiology or the disease. But when we started writing, and also now, the Flamin-go project, there was no organ on chip for the joint. So since our target organ is the joint, to try to do what we want, we're supposed to have a joint on chip. Now, just to go shortly into the detail of what we plan to do, this is the joint. You see, this is bone and cartilage. This is the synovial fluid. This is the synovial sheath with the synoviocyte, and the blood vessel, of course, important because the immune system can arrive into the tissue through blood vessels. So what we plan to do is to exactly recapitulate all these different tissue in a relevant way within the chip. So we will have a part dedicated to the bone that will be connected to the cartilage, that will be dispersed into this synovial fluid. Here we will have the synovial membrane, and then on this side and also in this one we will have a blood vessel, so really a tubular vessel-like structure in which the cells, white blood cells, will be fluxed. Now, as you may see in this different cross-section view, there is also under the synovial fluid a pressure that mimics the walking, because when you move the walking or the movement, let's say, of the joint, because when you move them, there is a stretch in this condition. And so the idea is exactly to try to see, when we put into this blood vessel new drugs, what happens to the inflamed synovia. I also put the concept because there are, let's say, some novelties in this project related to the fact that, let's say, 2 or 3. One is technological, because we will make this, since it is a complicated organ on chip, we conceived to produce it as Lego bricks. You know, Lego bricks, you have different bricks and you can build them together. So we will start by doing the blood vessel, the osteochondral unit, and the synovial unit independently, make them work, because they are also by themselves complicated, and then we will assemble them all together. Obviously, to give the complexity of each of this tissue, we will have a bioprinter, which is an instrumentation that allow you to put the cells in culture in certain disposition, in a 3D disposition. This is one of the novelties. The second one, very important, is that we will not start from, let's say, cells in culture, let's say cell lines, but we will start from patients' biopsy. So really, we will have from different patients, different biopsies, and so different organs on chip.
And I can already add a teaser for one of the next events. We will talk about 3D bioprinting as well, which is an important part of the Flamin-go project.
Yeah.
And there is one more thing here that I see on this slide with the concept at a glance. One is that you create this artificial environment, cells one after another, it's an organ, and you also need to somehow have a control of this environment or sense this environment in a way. And for this, as far as I understand, you use lab on a chip, right? Or like, yeah, what is...
No, it's true. Let's say that we use different ways. One is the lab on a chip. So let me just explain what a lab on a chip is. The chip is this form. Microfluidic means that the nutrients are continuously filled inside this chamber, like it happens with blood vessels that continuously take into our organs the nutrients. And so we will have nutrients going into the chip and nutrients that come out from the chip. This medium that comes out from the chip will also retrieve from the chip, like our vessel do, all lots of information, lots of mediators that meanwhile the tissue is producing. So what we retrieve from the chip will be analyzed on a lab on chip. This will be done automatically, automatically. And so we can have, let's say, a real-time advice of what is going on. For example, I give the drug and I can see if cells are dying, not dying, if they are producing some mediators or whatever. And this is one aspect. There is also another aspect. Let me see if I've got, This is, let's say, the technology that we are putting in our chip. So we are designing the chip because we need a new configuration. So we have got bioengineers that have drawn this channel in which we feed the cells. Then we've got the cells from the patients. We have this bioprinting. We will have a webinar on it. And then we have sensors. Now, what are microsensors? They are some particles that have some characteristic, I will tell you, that we put inside this culture together with the cells, and that will allow us to understand how the cells are performing. For example, are they good? Is the oxygen inside the cells good? Have they any problem? Is the pressure we are giving too much? So these are particles that mimic a cell, but for example, they are fluorescent, as you may see here. And let's say that if oxygen is okay, there is no fluorescence. If oxygen is missing, these particles start to be red. And so we can easily understand what is going on inside our chip.
It's really fascinating because in the beginning you started this story with where you work and what your center does and how you have all these different expertise of people and also patients there. And now when you explain the whole story, it really shows the importance of having all that in one place so you can follow through with everything, with the whole treatment.
Yeah, I think that this is really an important point. I stop my
you
sharing.
can
Yeah, I come back to you. Yeah, it is a very really an important point because having there all the expertise now. Obviously, we don't have the expertise now. The aim of the Flamin-go is to put together expertise that are dispersed into Europe. Each one, you know, we've got a group that is very good with bone and cartilage, another group that is very good in sensor, another group that is very good in doing the platforms, okay? We are, let's say, good in doing the synovia, and this will take the first 2 years of this project. Then we're supposed to assemble all the expertise at our center. And this will be important because we will really have patients there with the biopsies, and we will be able to make a sort of pipeline going from the diagnosis of the patient to how we can treat them. And there is a further step, because this is, let's say, a dream that I had while I was writing Flamin-go, you know, because we don't know if we will succeed. Obviously, research is, we try to do our best. We have an idea. We need to change on the way. But if we succeed, my dream is to start start a startup, because this will really take the results of the project to the market. Because, you know, the first and more obvious impact of this project is for the research. So we will demonstrate that joint on chip is feasible, that it works, that it can be done also by on patient sample. But this, okay, it's important if you work on joint, you know, it's important not only for rheumatoid arthritis, but also osteoarthritis, which is another big disease impacting a lot in Europe, okay? But I think that there are much more impacts that if we are able to take to the market will be important. For example, we can make a joint on chip, and it can be useful also for the pharma. You've got a new drug, instead of testing it in animals or in 2D layer, you can use this. It can be useful for the clinics, because in my mind, in 10 years, the hospital will use the personalized organ on chip to test the drugs, to make the diagnosis, you see? So I think that I hope that we will succeed in what we plan. It's very ambitious, but we've got lots of good experts from Europe, and so I'm confident.
Yeah. It's really nice. Like, first you have from all over Europe experts, and then you will try to bring it all closer and maybe do it on small scale, but then you also have ambition to basically scale it up for all the 3 million of patients who suffer from rheumatoid arthritis to have
that would be great.
That would be great. Yeah. It's definitely the personalized and precision medicine are the future probably of medicine. Hopefully we will get there. Thank you so much, Dr. Annalisa.
It's my pleasure
to take time today to speak to me. And as I said before, we will do more webinars as part of Flamin-go project. We will present the 3D bioprinting. We will talk about a lab on a chip and maybe some other things that are part of this big project and of these topics that deal with different organs on a chip and the future of medicine, as I said already. So if anyone, I didn't see any questions in the chat. I hope you enjoyed it. I hope you learned something new today, and I hope you will follow our projects also in the future. As Annalisa already said, we are now more in the beginning phase, so we have 2 or 3 more years to develop it, and I'm sure we will talk again with Annalisa at another webinar and present our your progress. You're doing most of the research. I'm just happy to be part of it and to communicate with others what's happening. So I'm looking forward to the next stages of this project. And thank you so much for taking time.
Bye-bye. Thank you very much.
Bye.